So really special senses were done in nervous system chapters, but in WCC, we do them here in A&P2 at the beginning. So in lab, we would actually have the models of the eye, and you'd be able to pretty much identify all of the structures there. We do a cow eye dissection. So this is more of a lecture, but I'm going to keep it to the identifying of the parts of the eye. So special senses, and special senses are anything that's not a somatic sense. If you remember from AP1, the somatic senses like touch, pain, temperature, vibration, those type of senses, tickle. Special senses are your vision, your hearing, your vestibular sense, which is a sense of balance, which is also in your ear, you'll see next week. The sense of taste, which is called gustation. The sense of smell, which is olfaction. So those are special senses, and they have a lot to do with the cranial nerves that are in your brainstem, that emanate from your brainstem, because the cranial nerves are actually peripheral nerves, but they have their nuclei within the brain. So if you remember the 12 cranial nerves, so with the eye, of course, the cranial nerve 2 is very important. Cranial nerve 2, and that's the optic nerve. And there's 2 optic nerves. So the optic nerve is going to take the signal from the eye and bring it back to the brain, the occipital lobe, if you remember. So there's 2 optic nerves, and that's cranial nerve 2. Special sense optic nerve. Not motor. There's no motor control of the eye with the optic nerve. That would be oculomotor and trochlear and abducens. So again, it's easy stuff just to identify the outer parts of the eye, and we'll look at the eyebrow, the eyelashes. I mean, these are really hairs, which we call cilia, and they're there for protection, not just to go to the salon and have them waxed and trimmed. These are important for keeping things out of your eye, because your eyes are very sensitive. And the term for eyelids is called palpebrae. So that's really going to be the answer on the practical. We use the word palpebrae. And we'll look at the different parts, the tarsal plate. Again, go to your checklist to get all the terms you need to know. Commissure, remember the word commissure is like the opening, really. And lacrimal, remember that word from A&P1? Lacrimal gland is tears. So lacrimal refers to tears. And tears are another line of defense against antigens or particles or dust, viruses, bacteria. So there's antibacterial qualities in the tears. You see tarsal glands along the core of the eye, below the commissure. Sebaceous glands, remember that from your integument. So very thin skin on the eye, but there is glands within that dermis of that integument around the eye. And the sweat glands as well, just like any other skin. So here's just the external stuff, and there will be questions like this on the practical. You have your eyebrow. There's not much we can say about the eyebrow. And your eyelashes, which are basically cilia. Right? So if this was the left eye, the nose would be here. So this would be more nasal side, and this side would be more temporal bone side. And here's the palpebrae commissure right here. And inferior eyelid, palpebral commissure medial. So medial is more nasal, and lateral is more temporal side of the eye. If this is her left eye, so you look at this left. How do I know it's a woman? I don't know really. We'll say this is the left because we know this is medial, right? If you're looking at the patient, that's her left. She owns the anatomy, or he owns the anatomy. The palpebral fissure is just the space, right? Just the space, the opening between the 2 palpebrae, which are eyelids. Conjunctiva, sclera, words like sclera will go into, and the white of the eye, just so you know, this is always a question, the white or the fibrous part of the eye is called sclera, the whites of the eye. The clear part that's in front of the pupil is called the cornea. So that opening right here, this little opening, is called the pupil. That's just an opening. It's not a structure, because the iris is the structure that's going to constrict or dilate. It's based on the sympathetic nervous system, actually, if you remember that. So the iris is this smooth muscle structure that has pigment, brown eyes, blue eyes, hazel eyes. The more melanin you have, the more darker your iris would be. So melanin is brown pigment, dominant brown pigment. So less melanin, lighter the iris, blue eyes, green eyes. So know in this external picture, that's really important. You know the pupil and the iris. This is a lateral view. This is a little tough one to see. I'm sorry, it's more of a medial view of the eye. And you can see with the light's coming in from the sun here into the palpebral fissure, which is the opening. And then the pupil is the opening behind that. So this part right here, you're going to see later, is called the cornea. The cornea is the fibrous outer covering. The white of your eye is called the sclera. And there's different muscles. If you remember the orbicularis oculi muscle that really has control over your palpebrae, your eyelids, upper and lower, in a circular way. Okay, so all these parts are important. This is a really good example. The pre-lab is really good. It's really what has everything you need to know in a more concise diagrams and pictures. This one's a little more intense. Like this is more from the textbook, more as if we did this in lecture. So this is good to get a deeper knowledge of what's going on in the particular PowerPoint that I'm showing you now. But the pre-lab is really good when you're doing your virtual labs and you're studying for the practicals. I think it's really important that you open up that pre-lab and keep it open as you're doing the exercises. It'll really help you learn this stuff over and over, because you're going to need repetition. So again, let's say this is a medial or mid-sagittal view of the eye, looking at it from the side. So some really important things. This is posterior eye, right? Posterior, back in the orbit. And you can see the branch of the optic nerve right here. This is all optic nerve with blood vessels flowing within it, and all the neurons that are taking the impulses from the retina back to the optic chiasm. Remember that, where it crosses over right below the hypothalamus there, above the pituitary gland. So the optic nerve is bringing the impulse back into the brain. It's really important, right? So here's the anterior. Here's where the light's coming in. All right? And so when you're studying the eye, another good thing to realize now, the light is coming in through the air, right? So the mediums change. The light comes in through the air, then the light has to penetrate the cornea, which is like a membrane. It's actually some epithelial tissue and some light connective tissue. So once the light gets behind the cornea, then we're into a fluid medium. And the fluid medium of the eye is called the aqueous humor. So the aqueous humor is more in the anterior chamber and the posterior chamber too. So aqueous humor is the fluid that's more in the anterior part of the eye, but it's back here too. And I'll show you changes. So it's kind of a clear fluid, a watery fluid. So the light comes through the cornea and then into the pupil. And once it goes through the pupil, remember the pupil is around the iris. The pupil is just an opening. And behind the pupil is the lens. And the lens will refract the light. And here's the lens. You can see right here. It's like normally it's like a football-shaped biconvex structure. It's protein structure. It's crystalline protein. So now after the light goes through this fluid medium, it comes into now a crystal medium, like a lens, where it's going to be refracted. So it's going to be refracted to hit a very specific spot on the retina. So then the light, as it comes in, will go through kind of a thick gelatinous fluid, and we call that vitreous humor. So vitreous humor is behind the lens. And really, the vitreous humor is that thick gelatinous fluid type substance that keeps the retina in place. We'll go through all these parts. Now, the retina is where you have your photoreceptors. Now, you remember receptors from AMP1, how they pick up the sense, the stimulus. The stimulus is the light, right? Remember from the sun? The receptor are in the retina, and there's two types of receptors. You have rods and you have cones. Rods are more for dim light, where cones are more for color and light or bright light, brighter light. So rods help us see more in the dim light. Like if you see in a sheep, they really don't care about light so much, but they really need to see in the dark. So they have different layers within their eye, and they have more rods than they do cones. So the sclera is the outer part. Remember, the white of the eye. So let me go through the three layers, and it'll go through there. Actually, it'll go through the next couple of pages. And if you see these vessels, you have veins are blue and the arteries are red within the eye, because you need a lot of blood supply to this tissue. So the three layers of the eye, the outer layer is more of a fibrous layer, and that's connective tissue. And that would be your sclera, the white of your eye, and your cornea. Your cornea is clear though. And then you have a next layer in, is called the choroid. And this is going to be the ciliary body. You'll see later on the ciliary muscle, the iris is choroid, and blood vessels. So it's more vascular. A lot of the vascularity, all these arteries and veins are part of the choroid layer, and the ciliary bodies. The ciliary muscles, which help the lens stay in place and also help accommodate to different vision. Like your lens is going to change shape. Like I told you, it's like a football shape biconvex. But if you have to see further away, it's going to change its shape to more concave. And this lens is really the reason that we need to wear glasses if we have near vision, which is nearsightedness, which is called myopia, or far vision, or farsightedness, which is called hypermetropia or hypermyopia. So the lens has a different position, a different shape, so that changes where the light is going to be refracted to. So the inner part is more neural, and that's the retina. Neural, that's where the nerve tissue is, in the retina. Now, the retina is a layer. Like if you look at the back of the eye, it surrounds the eye, and all of this has rods, many rods. There's much more rods than there are cones as photoreceptors in our retina. Now, in a place called the macula lutea, that's an area in the retina that has a spot called the fovea centralis. So this is an important area to see, fovea centralis. And the fovea centralis is an area with a high population of cones. So we want that light refracted from the lens through the vitreous humor right onto the fovea centralis. That would be the best vision, the most acute vision, the most detailed vision, best to see different colors and be able to differentiate colors. But right here, there's something called the optic disc. And what the optic disc is the area where the optic nerve leaves the eye. And there are no photoreceptors there. None, no rods or cones. So this is your blind spot. So the optic disc is your blind spot. So of course we don't see our blind spot. There's an an exercise, you'll see where you put a little dot and a plus sign, and you could cover one eye, and you kind of concentrate on the other, on the plus when you're going closer to it, and you'll see that that plus sign will disappear. That's the only way you can see the blind spot, is if you cover one eye and do that exercise. It takes a little while to get it, but your brain accommodates for that. Like, you'll never know anything about a blind spot unless you do that exercise. Don't worry about these layers. I know this may show up this is layers of the retina and the choroid within. Don't worry so much about these layers. I won't use these questions because you have enough to worry about. Now, lacrimal is the actual tears. So the tear glands, the tear ducts, the nasolacrimal duct. So tears, really, again, for humans, tears are strictly for protection. Again, I know we cry during certain times, emotional. So there's a limbic part of it as well. But really, in evolution, tears are more for your protection of your eye. So tears contain water and sodium, of course, salt. But there's antibodies, right? There's antibodies, viral protection. You have lysozyme, which is a destructive. Remember, lysosome from the cell was the suicide sac of the cell, which destroys something that's harmful to the body. So you have an antibacterial part of the eye. And the enzyme is called lysozyme. And of course, lubrication, cleansing, all of these functions of the lacrimal fluid. Moistens and reduces friction, but really cool that it defends against microbes, and that's really the reason that we have tears. And again, 15 per 20 per minute blinks. This is how many times you blink. It's like a windshield wiper fluid, so lacrimal. So here are the structures, and this is pretty much it. Look at where the lacrimal glands are. They're actually lateral, right? Or temporal. This is her right eye, or his right eye. So here's where the glands are. And these are stimulated, of course, by some type of irritation to the eye, to the cornea or the sclera. Then you have the lacrimal gland will kind of bring the lacrimal fluid across the eye like a windshield wiper, but except from the top down. Right? Puncta is just an opening. Caruncle is just that piece of connective tissue that's you kind of feel that. If you feel your eye, you feel that little hard piece of connective tissue that kind of helps set the eye, forms the duct there. So the puncta is actually a duct, or a hole, really. And canaliculi is a small canal, remember, in bone. So these little canaliculi drain into the lacrimal sac, which is right here along your lateral part of your nasal cavity. And this all drains through a nasolacrimal duct. You know, when you have tears, it can get into your pharynx. Actually, you can drain back into your pharynx and go into your throat, or out your nose, of course. So the tears will drain into the nasolacrimal duct and then out the nasal cavity. So know these parts. Of course, you know the nasal cavity. We'll talk about that when we're in respiratory. The nostril, or nares, is the opening. So lacrimal gland is two parts. You have your eyelid part, the palpebral part, and the orbital part. They're right pretty much on top of each other. But know that the gland is here. And then you have your lacrimal puncta, and you have your lacrimal sac, and the duct. Yeah, I think the hard part is getting gland, sac, and duct. Get those right for the practical easy points there. So again, I think there's a couple of that. I don't know, actually. I don't know if there's a lot of questions. So there might be a couple of questions in the virtual labs on lacrimation, which is tear production and secretion. So this is pretty much preparing you for the lab part, or the dissection part of the lab. So these just things, you have to really go through this because you need the definitions. Like the posterior cavity, by definition, is right behind the lens. The anterior cavity is in front of the lens, and that has aqueous humor in the anterior cavity. And by definition, you could say behind the lens is the vitreous humor. That's the gelatinous fluid. Humor just means fluid. And aqueous humor is more watery, like aqueous, watery. So watery in the front and gelatinous in the back of the lens. But there really is aqueous humor behind the lens as well, but it's okay. Behind the lens, we'll say it's all vitreous humor for all the way back. Here's the three layers. You have the fibrous layer, which is the sclera and cornea. The vascular layer is the choroid. And the retina is the inner layer, and that's the neural layer. Know that. That's important to know. That's like the kind of questions we'll ask that aren't identifications. And again, this PowerPoint will help you look at the way the visual axis. You can see this is a superior coronal, superior section, cross section, transverse section, looking down the skull. Remember the skull, remember the cribriform plate here, and the crista galli of the ethmoid. And here's the orbits. So this is left, and this is right. So you can just see where the light comes in through the cornea. Remember the mediums change from air to fluid to gelatinous material, and it's all refracted from the lens. This is really good. This is why I kept this in here. I know there's a lot in this PowerPoint, but the pre-lab, again, is really great. I'm going to show you that before you go on and do your lab. So the outer part, again, fibrous tunic. This is kind of memorization now. And you can see, again, this is the eye from the mid-sagittal section. So you can see the light coming in from here. This is the anterior. Remember, this is the optic nerve, right? CN2. Optic nerve. You can see the white of the eye is up here is the sclera. The anterior transparent area is the cornea. Then you have your aqueous humor, which is all in here in front of the lens. Everything back here is vitreous humor. Here's your lens, which again is biconvex. And then you have these ciliary bodies. Now the ciliary bodies are part of the choroid, right? The choroid is all here, vascular. So the vascular tunic means layer, by the way. And you have these ligaments that attach this to the ciliary body, and the ligaments attach the lens to the choroid. Here's this ligament. So what the ligaments do is they either tighten or loosen based on what shape you want the lens to be, whether you're looking at something close or you're looking at something far away. And that changing of the lens shape is called accommodation. And again, that could be distorted if you have a problem with your eye. So we'll go through a couple of the problems you could have with your lens, like astigmatism, which is a change of shape of the lens or the cornea. So these suspensory ligaments or ciliary ligaments, they have different names, we'll see what your book calls them, are attaching the choroid to the lens to help it change shape. And here's the retina. This is the important part. You can see how it is continuous with the optic nerve, because it's going to send a signal through depolarization back to the occipital lobe to see what color you're looking at, what I'm looking at, memory of something I've seen. Vision is a very big special sense in the human brain. A lot of it actually takes up, I would say, 30% of your whole brain is just tracks for vision control, especially sensory. So here, this is a little more detailed. You can see the three layers again. Go through all of these. Make sure you know the anterior posterior chamber and aqueous humor. All of this, now you have to picture all this in here is vitreous humor. When you do the dissection or you look at a dissection, I'll put up one of my dissections into this learning module so you could actually see the gelatinous quality of a vitreous humor. It's really like a big ball of gelatin as opposed to the anterior, which is all the aqueous humor. So read through these, you get a little more information, what each is made up of. Right? The sclera is dense connective tissue. Like if you ever did a dissection of an eye, you see it's kind of hard to cut through the sclera because it's so dense connective tissue. And it gives it the shape, that roundish shape. Sometimes it's a little oval shape, and that changes your vision, the eye. And it's, of course, protection. Right? The cornea, of course, is transparent. So it's got some collagen connective tissue, but it also has simple squamous epithelium. It's important. Of course, we're not breathing diffusing a lot of air or oxygen in our eye, but there is oxygen that can get into your eye, and that helps the eye function. But of course, we're not relying on getting oxygen through this area of our body. Of course, this happens in the lungs. Okay, and the outer layer is more stratified squamous, so that's, is very mitotic. If you ever want to get into a really good field, ophthalmology is a really great field. A lot of new technology, and where they're going with vision is very amazing. Okay, so corneal epithelium, outer, is more stratified squamous. High mitosis, changing over a lot, again, provides protection against friction as well. But sclera, much more dense connective tissue. Cornea, more epithelial. And there's a junction between sclera and the cornea called the limbus. I don't know if that's on your list. That's good to know. So refracting light. So remember, light comes in from the air to the cornea, which is all these things: simple squamous, stratified squamous, epithelial tissue, really. Remember the big four tissues, right? From AMP1, you have epithelial tissue, you have connective tissue, you have muscle tissue, and then you have nervous tissue. So the eye has all of them, and that's why we call it an organ system, because it has all the tissue types. Just like the skin, which you learned, and the muscle system you learned in the nervous system. So this goes through all of them. This is really good. All the different tunics, what it has. So again, this PowerPoint is much more detailed with definitions. It talks about the ciliary body with the muscles, and they connect to the lens, which connects via the suspensory ligaments. Again, these are all important checklist things for your practical, and the ciliary processes. So basically, the ciliary muscles control the shape of the lens to refract light better onto the fovea centralis, which is part of the macula lutea, which is part of the retina. So within the choroid, again, this is vascular, this is part of the choroid, is the iris, and that's the smooth muscle part of your eye that has color. Right? So you have different reflexes, like you have dilation, and again, that would be dilation would be sympathetic, right? When the bear is chasing you in the woods, your pupils dilate, right? When you are eating or you rest and digest, then you have more contracted iris. So that's more parasympathetic. So I like these kind of questions, so these reflexes. Then the pupillary reflex, like if you shine a light into someone's eye, then it's going to be, normally the pupils are going to constrict to protect the retina, really. So if there's a problem with the sympathetic nervous system or parasympathetic nervous system, then your eyes won't constrict, your pupils won't constrict, they'll stay dilated. Or if like when you go to the ophthalmologist or optician to get your eyes checked, they put in a dilator, right, which is atropine, which stimulates pupil dilation. So they can look into your eye easier. So every time they look into your eye with the light, they won't constrict your iris, so they can see back into the retina, which is really cool. And the eye actually is the only place where you can look into the body from the outside and see the retina. You can see nerve tissue. You can see the optic disc. And there's going to be a picture here sooner or later of that. So here's the diameter of the pupil. You can see like it's small here, constricted, more parasympathetic when light shined into it. Now this is dilation. You can see the pupil is wider, allowing more light in, and you can see how these muscles work. You have these contractions, these papillary, these more radial muscles, and the more circular muscles on the sympathetic side. Change the opening of the pupil, really, well, changing the size of the pupil. Pupil, remember, is the opening. So here's the retina. Now this is, of course, the most important part for our vision, our acuity of vision. And this is where we really want to protect. It's funny because the retina is really not a stable structure. It's actually held back in the back of the eye against the choroid by the vitreous humor. So you really want the correct amount of pressure in your eye, like excess pressure. I don't know if you ever heard the term glaucoma. Is too much pressure in that vitreous humor and aqueous humor too. So that's excess pressure. It's a really dangerous condition because it could throw off your retina and affect your acuity of your vision. It could lead to blindness, of course, ultimately. So the neural layer is really the main part. That's where your photoreceptors are in your retina. And your photoreceptors are your rods and cones. Remember, rods more for dim light, cones more for brighter light or color. Cones are for color. So somebody who is colorblind, which is more common in males, you have a less amount of pigment in some of those cones. Like you can't see green or blue. You can't differentiate for green or blue. It's more about the cones, the pigments that are in the cones, because rods and cones have pigments in them. Again, we're not going that deep into phototransduction, but at least you have to know those terms, rods and cones. And ora serrata is, and when you do dissection, you can see that's a kind of a structure that you can see from the sclera. That's actually part of the retina, but it's non-photosensitive. So you'll see a picture of that when you do your lab. Just another thing. So deep in the retina, you have these layers. Again, I'm not going to hold you to these layers. I want you to know about photoreceptors, not so much the bipolar layers. These are the types of neurons and the ganglions. Of course, remember, ganglion is a cell body that's outside the central nervous system, a peripheral, a bundle of cell bodies. So you don't have to know all these specific things, even if that's on the list. But you can see, like if you're looking at the retina here, if we took this retina out and extrapolated it into a larger structure, you can see how there's layers. Here's the choroid out here. And the retina, of course, is deep to the choroid. You have to know those terms. And the light, as it comes in, goes through, back, and then comes back and goes to the optic nerve. So you have these layers of cells, and these are all neurons that transduce and send the signal from, here's a rod right here, cone. And that's the photoreceptor layer, right? Rods and cones. This is important because the back of the eye, you can see there's an optic disc. And this is really where the optic nerve exits the eye. And that's your blind spot because there are no photoreceptors on that blind spot. Macula lutea is an area of the retina that contains the fovea centralis. That means central pit. So the fovea centralis is really the important area where most of your rods and cones are, especially cones, right? Yeah, your cones are really about vision, differentiation, what you're seeing, the sharpest vision or acuity, visual acuity. That's really what they test for when they use that Snellen chart, which is part of your virtual lab. You'll see how to use the Snellen chart to decide if somebody has perfect vision. Perfect vision is 20/20 vision. So somebody at 20 feet could read a certain line on that Snellen chart, those letters, and somebody who can't would reading the same at 40 feet that somebody with 20/20 vision could see at 20 feet. And you'll see how that works. It's very simple. Here's the blind spot. This is really cool. Like this picture right here is what your doctor will look into your eye with an ophthalmoscope or a fundoscope. And you're actually looking at the retina. You could see the blind spot. You can see the blind spot here, and that's the optic disc. You can see blood vessels. You can see that little depression there. That's the macula lutea, right? And inside the macula lutea is the fovea centralis. This is a perfect view. And again, like I said, this is one way you could see nerve tissue and blood vessels from outside the body by looking in through the pupil. And optic. So know this picture. Know this picture that which is and really know the difference between the fovea centralis and the optic disc. So when you look into somebody's eye like this, you could actually see if they have things like atherosclerosis because it affects the blood vessels. You possibly could see if they have diabetes because of the destruction in the blood vessel look. They could be wiry or puffy or woolly looking. This is a very normal, obviously normal fundoscopic or ophthalmoscopic view where everything is normal. You can see the blood vessels are normal, but if you see any changes, this could be a sign or definitely a late sign of diabetes, atherosclerosis, which is fatty plaquing of the arteries. So it's cardiovascular disease, which you'll learn later in the semester when you do the cardiovascular, when you do the blood vessels. So this right here is the chart you use when you're trying to locate your own blind spot. Like, I guess, I don't know if you could do it by looking at this small of a picture, like there'll be a bigger one. And you're going to just draw this, like draw this on your, on a whiteboard or draw it on your computer, and then try to find your blind spot. You cover one eye, you focus on one part, and you move your head to and from the image. So you can see if that plus sign disappears or this dot disappears, then you found your blind spot. So practice that when you do your virtual labs. Okay, so you have some diseases. Again, we're not going to talk about retinopexy. It's kind of cool to know about near vision, far vision, glaucoma, what cataracts are. So I want to see if they have anything on there. This is talking about the lens changing shape. Again, here's a photo. When you're looking at far, the way the lens changes shape. So if you're looking far away, the lens gets more convex, it gets more biconcave, fatter football when you're looking at something close like your watch. So as you go through your exercises in the virtual labs, you can see how that accommodates. It's called accommodation. Hopefully I'll spell it right. So accommodation of the lens, and you have a lab for that. So this will be some question. There might be a question about accommodation. Anything that's in the virtual labs. Again, I know there's a lot of them in this first lab. It's good practice. And then once you get to the second, third, fourth lab, this will be so easy to do, and you'll be able to manage your time easier. Okay, so of course you get a problem called cataracts, is a opacity in the lens. So the lens can't refract the light correctly because it gets cloudy. So cloudy is opaque, harder. So nowadays they actually just replace the lens with a new lens, a fake lens, and that cures the cataracts for the old. It happens mostly in older people. I'm sure if you ask your parents or your grandparents, they'll tell you they have a cataract, which is an opacity of their lens, and it's usually the result of just time, aging. Emulsification, big fragmented lens. They use ultrasound to try to cure it, so they don't have to do surgery, where they don't have to go out and take phaco emulsification, where they don't have to actually remove the lens. So that's a treatment maybe for somebody younger who has a cataract. And here's talking about the humors. So go through this. Again, vitreous is more gelatinous, and behind the lens is your definition, book definition. Again, it supports the retina. So really the retina doesn't have its own connections to the choroid. It actually has to be pushed back by the vitreous humor. And look at the dissections, look at the videos, you'll see what that looks like. So aqueous humor, I will say that's in front of the lens. Okay, and that's important for transduction of the signals. And this sclerovenous sinuses are important because it drains the fluid. Like, we need to drain that fluid. It can't build up in those chambers. So if you have too much aqueous fluid building up in the anterior chamber or posterior chamber, you're going to get what's called glaucoma, which is high pressure in the eye, which really could affect the retina position and cause blindness. So glaucoma is increased pressure in the eye. And they'll test for that when you go to the doctor. It's a tonometer. They actually pop up like a air onto your eye to check the pressure. I think the average, the normal pressure is 15 millimeters of mercury. Anything above that actually is dangerous. So they would have to drain your eye if you have glaucoma and treat it with drops. And it's really something. It's a serious condition that can lead to blindness. So glaucoma is increased pressure in the eyes. And it talks about this right here. You don't have to know the different types of glaucoma. Just read through so you know it when we ask on the practical question what glaucoma is. Okay, so here's the vision types. You have emetropia, is normal vision. You have to know this. Hypertropia or hypermetropia. Hyperopia is farsightedness. So you have trouble seeing close, but you can see far because of the shape of the lens. So the lens is more concave, so you have to treat it with a convex lens. And that's your glasses, right? That's your glasses. Myopia is you can't see so far away, right? So you wear glasses, so you wear concave lens glasses. So you're nearsighted, right? So farsighted as you get older. is a problem too. You tend to get more farsightedness, right? So this is corrected with the convex lens. And age-related change in vision is called presbyopia. You might, and this happens in young people like you guys, you get astigmatism is a curvature or change in shape of the cornea or the lens, and that affects your vision. It can cause headaches, and a lot of times you get glasses, prescription glasses, to treat that astigmatism. So astigmatism is change in the shape. Okay, and this goes through all the things. This can help you do your lab. I just want to go over to the pre-lab for you before you go. Again, this is you don't have to know this particular thing. Of course, you're going to know all of these things, all of these parts of the eye. And this one you might have to remember the midbrain, the corpora quadrigemina, superior colliculus. They're involved in visual reflexes. The lateral geniculate of the thalamus involves visual reflexes, and as opposed to auditory. Now, these I think are really important. When you go through these PowerPoints, look at these tables, because it's really good review. And go through all the parts. You have everything. You have everything you need to know about those parts. So keep these here if you ever need them. And there's some practice on here too, where you can practice looking at the muscles. And here's the dissection. Now here's the eye. Of course, now everything looks crazy now. When you're looking at a sheep eye or a cow eye. And you can see this is the cornea because the lens is behind that. I can hear you see now this is what the lens looks like. So here's the optic nerve. And this is important. This tapetum lucidum, just wrote over it. Tapetum lucidum, we don't have that in humans. That's more in other mammals like cows and sheep bovine. So the tapetum lucidum is in the choroid layer of their eye. It has like a turquoise look. It's pretty cool, tapetum lucidum. And this helps them see better at night, reflecting and harnessing the light. We do not have that. But we do the eye dissection, kind of remove the choroid here. This is all choroid, and part of the choroid, if the retina, of course, is very delicate, the retina kind of falls off very easily. This is just like a torn up retina. And here, really, you can't really even see it that well. It's so thin, when you see it. Choroid's thicker, and of course, the sclera, the fibrous part, is much more dense connective tissue. Right? And you see the lens after you cut this. Of course, you cut the cornea off. The cornea is gone. If you look back here, you can see the cornea there. And these are all muscles. So the eye, and I don't have it yet. I could put my own dissection of the eye here. And here's more tables where you could practice this is what you'll be doing. Like you'll be doing the visual acuity test. Is that Snellen chart with the letters. And then you have the accommodation, which is your lens. You see if you accommodate to find out if you're more nearsighted or farsighted. This is the blind spot chart where you can actually see where the blind spot is. It's kind of hard the first time. I think it takes a while to figure out. I mean, not that long, but you could actually keep doing it and you'll find. So this page, actually, you can do it. If you cover one eye, and in the lab, it'll show you. This is for color blindness, test for color blindness. It can actually show you the different colors. If you could read the numbers or letters, then I'm sure somebody, or not everybody, but most of you have had an eye exam. So you guys okay? You guys with me?
Yeah.
Yeah.
So that's basically the breakdown. I guess it's a little intense, but I just want to show you the pre-lab before you go and do this lab. Let me just show you what the pre-lab looks like. I just have to get that up. Give me a minute. So this is the pre-lab. This I really like these a lot. So I would spend a little time in here before you do the lab. And it gives you all the parts. You can see this the right eye. You can see the muscles. And of course, you have to know the muscles, the superior, inferior, lateral rectus, and the two obliques. 6 muscles of the eye. That's kind of tough too, because you have to know if it's right or left eye to see which muscles are which. So this is embedded in Blackboard. So here you have again the mid-sagittal. This is better than the PowerPoint because it's more descriptive. You can see closer up, you can see the ligaments, you can see all the parts, the sclera versus the cornea. And then you have the cut model. And then you see the blind spot versus the macula lutea or fovea centralis. You can see the layers. That's a good start, I think, when you really learn the layers, the fibrous layer, the vascular layer, and then the retina. Here we go. Here's more anterior from the inside. So now you're looking from the back of the eye out. So you can see the lens there. This is not the front of the eye. It's like looking from the back, from the inside, looking from the inside. You can see the ciliary muscles, which are part of the choroid, the lens, and you see part of the iris, not into the neural layer yet. So this goes on, again, seeing basically what we just did, more pictures, which are really what we need to know what that looks like. And now you have a dissection. So again, you're going to do the dissection video, which you see this is all happening, how it's cut. You can see the different tissue. There you go. While you guys are doing this, I'll send over the video so you can see me doing it with other students. Still see the pupil now. There's the lens taken out. Close up of the cornea. Of course, this is a dead structure. This is a dead sheep or dead cow. So the lens, the cornea is going to be kind of lousy. You know, it's going to be not look so good. So you got to remember it's a dead specimen that's been preserved. So let's keep going here. There we go. Let's see the same pictures kind of over and over. A lot of repetition in here. I just want to see if there's anything in here that, oh, here you go. This is a little deeper. He sort of shows you the difference between the cone and a rod. And these have photopigments. Now remember, rods are more for dim light, black and white, whereas cones are more for brighter light and color. And fovea centralis is mostly cones. See the gelatinous vitreous humor, which is on the bottom there. You can see it's kind of gelatin-like. And again, going through all the things. Oh, here you go, the lacrimal apparatus. So I really like this one. I think it's one of the better study aids, I think, from McGraw-Hill. Here's your Snellen chart, which again, you could practice that. And I guess, you know, you could do it with somebody, you know, that you're at home. If somebody doesn't mind being a subject for you, you could test their vision. And here's the check for astigmatism. The chart, I think it's part of the lab. I think I left the astigmatism one on there. Accommodation of the lens, focusing on distant versus nearby object. And the shape of the lens changes when you focus on them. That's really important. Okay, near point vision, that's on in your virtual labs. Color blindness, I believe I left on there. That's kind of an easy one. And correcting the problem, whether you have hyperopia or myopia, and what correction you would do for glasses. So giving them concave lens or for or convex lens, whatever. Okay. I don't know what happened to my video. Hold on a second. Let's go crazy. You guys hear me?
Yeah.
Yeah.
Yes.
Yep.
Did I miss anybody in attendance? Frederick, are you here? Kenya, are you here? Okay, that's cool. So we got everybody else. I don't know what's going on with my video. You can't see me, right?
No.
No.
Yeah, I don't know what happened. Let me try to put in my webcam. See if that works.
My light is on, but you can't see me. Oh, there you go. Now it works. Hi, everybody.
All right.
Hello.
So that's about the way we'll do it. Now you're kind of on your own for the virtual labs. Are you okay with that?
Yeah.
Yeah. So you have a week to do them. So, get it done as soon as possible. You don't have to rush. But again, like I suggested before, I would go through the labs, the videos and PowerPoint if you want. I really like the pre-lab for today. That one really covered everything I think that's on our checklist. So do you have any questions about the eye? Okay. So what we'll do is I'll hang around till about 9:45 if you have any questions. I mean, I suggest you start them now since I'm here, but I mean, it's okay. You could do it and email me later. You don't have to do it now, but you have to do the virtual lab before 10:50. Everybody okay with that? Unless you don't have McGraw-Hill yet, then you'd have to let me know that. And I'll see it. I'll see who doesn't have it basically later on. I'll double check and I'll open it up to you. Okay.